antigen to blood that has antibodies against that antigen. Okay, so if I'm B, I can't be added to any blood that has A antibodies. Okay? Oh, here we go. If I'm B, I can't add any blood that has, if I'm B, I can't add to any blood that has B antibodies because it will destroy every single one of the red blood cells that I add in. So if I'm type blood B, I can only be added to B. Okay. Can I be added to AB? Can I donate? Can A be donated to AB?
Yes. There's no antibodies against it.
So, are there any antibodies against A in here? No. So I can add to AB. Are there any antibodies against B here? No. So I can add to AB. Are there? I can already add here, and I can add here. Okay. Now, who can I receive from? Now, this is looking at it from the other side of the mirror. I'm the blood receiving the donations. I can't receive blood from anybody that I have antibodies against. Okay, so it's just the same concept, but the other side of the coin. So if I am blood type A, I can't receive blood that has antibodies A. All right? Let me rephrase that. Let me rephrase that. If I'm blood type A, I have antibodies B. I can't receive any blood type that has the B antigen. I can never add the antigen to the antibody. So if I am blood type A, and I have antibodies B, I can't receive anything that has B blood type, B antigen. Okay? So if I am blood type A, I can receive from A. I have B antibodies, so I can't receive anything that has a B antigen. So can I receive blood type B?
No.
No. Can I receive blood type AB?
No.
No, because my B antibodies will react against that. Can I receive blood type O?
Yes.
Yes. Okay? If I have blood type B, I can definitely receive myself. I can't receive A because I have antibodies against A. I can't receive AB because I can't receive AB because I have antibodies against A. So, O again. Yes.
In AB, you don't have antibodies against A.
No, I'm receiving AB.
Okay.
I have antibodies against A. Remember, I can't add antigens to antibodies. I have the antibodies. So if I'm blood type B, I have antibodies against A. I can't receive AB because I have antibodies against the incoming A. Okay? So if I am AB, remember, I can't have antibodies against anything. Do I have any antibodies if I'm AB? No. So I can receive everything. Does that make sense? Okay? So AB can only donate to AB because it has both antigens. O has antibodies against AB. A has antibodies against B. B has antibodies against A. So no matter who I donate to, they have antibodies against me. But now that I'm receiving, if I'm AB, I don't have any antibodies. So I can receive anything. Okay, so I can receive AB, I can receive A, I can receive B, and I can receive O. Okay? Now I'm O. I have antibodies A and antibodies B.
Oh.
I can only receive O. Okay? Now notice, all the ones that donate, every blood type can donate to AB. AB can receive every blood type, okay? O can receive, all the blood types can receive O. O can donate to all the blood types. So you can donate to your blood type plus AB. All right, you can receive from your blood type plus O. It's not another rule. The easiest rule to remember is antigens cannot be added to blood that has antibodies against them. That's one rule that will save you. If you think about it, we'll answer every question. But you can double check with this. All right? So you don't want to add antigens to antibodies because every single red blood cell will get clumped. That's the key factor. Now, what antigen have I left out? D antigen, also known as the Rh factor. All right? So back in the early 30s, 40s, and 50s, doctors were noticing that babies were being born already died or dying. And when they looked at their blood in the microscope, the red blood cells were lysed and were blown open and ruptured. And so they started mixing different blood types together, and they came up with the ABO blood types. But even when those blood types matched, babies were still being stillborn. And so in the 50s, further research was done. It involves rhesus monkeys, hence Rh factor, and prisoners from a prison in Nashville that got release time or better food or whatever for being part of a scientific experiment. And the Rh factor was discovered, also known as the D antigen. But instead of saying that somebody has ABV blood, we use positive or negative. All right? So a person's blood type is A negative. They don't have the D antigen. And the interesting thing about when they do have the D antigen, and we call them A positive, hence their blood has both A and D, we still don't change the antibody setting. Because unlike the ABO, the D antigen is like our typical antigens. You don't get antibodies unless you're exposed to that particular antigen. So if your blood type is A negative, you're not going to have antigens against the Rh factor unless you're exposed to it by getting our A positive blood. Okay? So I'm not going to change any of the antibodies. So B negative has B antigen. B positive has B and D. This table is in the lecture notes and on the slide. B and D. AB negative, nothing different. AB positive has A and B and D. O negative, nothing different. O positive has D. But notice I didn't change the antibodies. All right? So if I tell you that we were looking at somebody's blood and A and B antigens were present, what would their blood type be? AB. What if I told you that A and B and D antibodies were present? No. A and B and D antibodies.
O negative.
O, because of the A and B antibodies. D, the negative, because they were negative and were exposed to the antigen and developed antibodies. Does that make sense? Let's talk about it again, okay? So they were O negative, so they had antibodies A and B already. They had a transfusion of O positive blood and developed the D antibody. If they were already O positive and they had a transfusion of the positive blood, they wouldn't make any antibodies against D, okay? So now let me say that they have no antibodies. What's their blood type? AB, question mark. AB, negative and positive, doesn't have any antibodies. So they could be AB negative that haven't been exposed to AB positive, or they could be AB positive. So if there's no D antibodies, listen carefully, if there's no D antibodies, then you can't determine anything about their Rh positive or negative. Because they're born without antibodies if they're Rh negative, and if they're Rh positive, they wouldn't have antibodies anyway. So it's only when they've been exposed to the D antigen and develop antibodies against it that you can say for sure that they're negative. Okay? So if I say, what's the blood type of someone who has B antibodies? A question mark. A positive, A negative, you don't know. Okay? But if they had B and D antibodies, then it'd be A negative. All right, because they had developed the antibodies. All right, now we're almost done here. Negative is like a cipher. It's the absence of something. All right? So it's not going to change our transfusion one way or the other, as far as who we donate to. But if you're negative, you don't want to receive positive, because it could trigger those antibodies we were talking about. Okay? And we don't want to do that. We don't want to make somebody make antibodies against the D antigen if they don't have to. Very critical if they might be a mom. Critical for a guy, because they might get a transfusion by accident that has the D antigen. If they have the antibodies already, then they're in trouble. Okay? So if we are negative blood type, we don't want to receive a positive D antigen. And if we are positive blood type, you don't want to be given to a negative type. So let's look at A negative can donate to any positive or negative blood type. Okay, it's just like A. The negative is an absence of something. So A negative can go to A positive or negative, AB positive or negative. Same with B negative. It can be donated to B positive or negative, AB positive or negative. AB negative can be given to AB positive or negative. All right? Now, A positive. We only can give to positive, can only be given to positive. All right? If we give it to negative, we're going to trigger that immune response. So A positive can only be given to A positive or AB positive. B positive can only be given to B positive or AB positive. AB positive can only be given to AB positive. And O positive can only be given to A positive, B positive, AB positive, or O positive. Right? So we can only give positive to positive, negative we can give to either or. Now again, the other side of the coin, receiving. If I'm negative, I can only receive negative. If I'm positive, I can receive either. All right? So A negative can receive only A negative or O negative. A positive can receive either. Okay? So what blood type is a universal donor? O negative. That's my husband. Everybody wants his blood. We get 2 or 3 phone calls a month from the blood bank. Not because there's that many people out there that have no O negative blood, but because it can be given to anybody. Okay, it can be stored in the pantry, the blood bank, and anytime they run low of the actual matching type, they can use O negative. So this is the universal donor. What's the best blood type to have? Because you can receive anything. AB positive. Notice how selfish AB positive is. The only people that can receive AB positive is AB positive. Okay? Look at poor O negative. Well, if my husband needs a transfusion, the only person that can give him a transfusion is O negative, and I'm O positive. As are all my kids. Okay? So the most unselfish donor can only receive one blood type. All right? Now, just a short discussion. There's a little bit of activity on PhysioX with this. When you come back on Thursday, we'll play around with it some more. But there is with ABO incompatibility and with this RH incompatibility, there is this disease. Erythroblastosis fetalis, not fatal fetalis, it is fatal, but it's fetal, referring to babies, okay? It is only a cause of concern when the mother is RH negative. If the mother is RH positive, this will never be a problem, okay? It will only be a problem if her baby is RH positive. But if her baby is RH positive and she's RH positive, it won't be a problem. So it's only a problem when she's negative and the baby's positive. If she's negative and the baby's negative, it won't be a problem either. Okay? RH. The D antigen.
Oh, okay.
Okay? Now, the only way the baby can be an RH positive if the mother is an RH negative is if the father is RH positive. Okay? Because this is a recessive gene. If the father is negative, the baby will be negative, or the husband wasn't the father. Okay? So, what happens is the first baby is usually okay. But at delivery, as the placenta separates from the decidua, the stratum bassalis, some of the baby's blood cells enter the mother's bloodstream. Now that first baby is in her arms. Baby's safe, okay? But the baby's blood cells in the mother's tissue are coming in contact with the mother's B lymphocytes. The D antigen is foreign, and the mother starts making antibodies against the D antigen. Is it going to hurt her? No, because she doesn't have it, okay? The next time she has a baby, if it's RH negative, will there be a problem? Nope. The next time she has an RH positive baby, these antibodies can cross the placenta and cross the amnion and start agglutination of the baby's red blood cells while it's still in utero. At first, they were only able to give these babies transfusions after they were born. And then technology developed in the 70s, and they were able to give the babies transfusions while they were still in the uterus. They'd send a needle into the abdominal cavity and give the baby a transfusion. But they were still losing babies. And so a process was developed by which if a baby was born and it was tested, they knew the mother was RH negative and the baby was RH positive, the mother had to get an injection within 72 hours, 3 days, of a material known as ROGAM. This stands for immunoglobulins, IgG, against the RH factor. Passive. You didn't make the antibodies. So what's the purpose of ROGAM? Why does it have to be given within 3 days?
Before her body will make the antibodies.
It takes 3 days to start the process of her body transforming the B lymphocytes into plasma cells and making antibodies. If those red blood cells of the baby with the D antigen can be destroyed before the end of those 3 days, the mother's body acts as if it never had them. And she doesn't make antibodies. Okay? So there was a case where the mother's first child was RH negative. Her second baby died of erythroblastosis fetalis. Her first RH positive baby. She never had a transfusion. The doctors were trying to figure out how all those antibodies ended up in her blood. And this was her first baby that was Rh positive. And what they finally decided was that that first Rh negative baby was a twin. Red blood cells appear before the end of the 3rd week, beginning of the 3rd week. They're there before the heart starts to beat. And that twin, a fraternal twin who was Rh positive, died. It was just, barely an embryonic disc. And as it died, the tissue was absorbed back into the mother's body, including those red blood cells with the D antigen. And she made antibodies against it, even though she didn't even know that she was pregnant with that child. It doesn't have to be a twin. It could be any pregnancy that the body aborts or reabsorbs and dies before the mother even knows she's pregnant. So now, to avoid that, women who are pregnant, only if they're Rh negative, are given the RhoGAM shot several times during the pregnancy, just in case. She had a previous pregnancy, or this one is positive, and they don't wait to find out if the baby is born. Okay? So again, she's not making these antibodies. This is like getting a snake bite, an anti-snake bite venom. Those antibodies are raised in a horse against the snake toxins and given to you if you get a snake bite, because you don't have time to wait a week or so for your antibody levels to rise. Okay? So they have to be given each time. They're not going to be saved in the mother. They're going to be broken down by our system, and they have to be given each time. All right? So we'll start with that, give you some time to absorb it and digest it, and we'll review a little bit on Thursday. The lab that we have with the antibody testing is not too long, but it gives you practice in assessing the presence of antigens. So what you're going to do is you have 4 different individuals. If you turn to the last page of lab 14A, which I think it says in your schedule 13 is due, but it's really this 14A page 13 is what I'm asking you to look on. It starts with page 11. If you look on page 13 down the bottom, there is a list, a drawing of blood typing trays right here. This is what we're going to do right now. Okay? And here's how it works. We're going to take 3 drops of fake drops of somebody's blood, and I can never, I swear, need to wear 3, 5, 10 of these red. Anybody see my red ink? That one is raw purple blood. Right in front of me, I swear. I said. So let's say we have Ms. Brown. I thought she was Mr. Brown, but she's Ms. Brown. And we're going to put 2 or 3 drops of her blood in each of these trays. Okay? In your case, they're actually shaped like this. And here's the 4 vials of the 4 individuals: Brown, Jones, Green, and Smith. Sorry. Imaginative names. And then to each of them, we're going to have a drop, add 2 or 3 drops of antibodies. Not her antibodies. I don't care what her antibodies are. I am on a fishing expedition for her antigens. Okay? So I've got chemically prepared antibodies against the A antigen. Antibodies against A. Antibodies against B. And antibodies against the Rh factor. And I'm going to use a separate toothpick for each one, otherwise I'll be transferring the antibodies from disc to disc. And I'm going to stir it up. Okay, whoops, do that. A different one, and a different one. Now, if that antigen is present, what is the antibody against A going to do to red blood cells that show antigen A? It's going to clump them. It's going to cause agglutination. And so I'm going to see, I see it now. It's going to cause those red blood cells to clump. It might look thicker. It might look like turn into gelatin. If you look at it in the microscope, you'd actually see this clumping. But let's say that's all that happens. The antibodies I added against B did cause an agglutination. And the antibodies I added against the Rh factor D didn't cause agglutination either. So what's the blood type for Ms. Brown? Do I have B antigen? Did it cause agglutination? No. Which antigen was present and was found by the antibody that I added?
B positive.
A. A is the one that agglutinated. So when I added the antibody A, it found the antigen, bound to it, and agglutinated the cells. When I added the antibody against B, it found nothing, which means my antigen is not present. When I added the antibody against Rh factor, it also found nothing, and therefore didn't clump any red blood cells. So the only antigen I have is A. My blood type is A negative. All right? Now let's say that this happened. What's my blood type? AB positive. Okay? And if this happens, O negative. Because the antibody found no antigen to bind to. That meant the A antigen is absent, the B antigen is absent, the D antigen is absent. When I have no antigens, my blood type is O negative. Okay? So, what you're going to be doing is, in groups of 2 or 3, grab 4 bloods, 3 antibody solutions, and 4 typing trays. This works best if you do it on a sheet of white paper. So you can use a paper towel. You can just be careful you don't mix one tube with another, so you can lay them out and put the names under each one. Add the drops, do one at a time, however you want to do it. And then record your results by drawing in similar to the way I drew it on the board and then identifying. Sometimes it's a little difficult to tell whether agglutination has occurred, so you might want to do several and get an idea. I'm more interested in that you can tell if agglutination is not crazy. The lab quiz or exam will be asking you to read the typing results, not has agglutination occurred. So I will show you on the lab exam or quiz image, and I will say this is agglutination, this is no agglutination. So you're not trying to interpret it. I will tell you what it means. And then you tell me what the blood type is after that. Okay? So when you're done with this, just rinse out your trays. They're not disposable trays. I'm going to put them back here. So, you need to get you're going to put back and get those. You can use one side of one, let's try the other. Otherwise, you'll need section 12. You want to repeat it, look at it again, you're welcome to do so.
Antibodies.
Right.
Yeah, so if it breaks it down, then it's that's what it is.
Right.
It breaks down to it breaks down the B, that means it's positive.
What we're going on not worry with this test, what antibodies are present. We're using antibodies to get that report. You need to be down there, put in there and see what antigens we find. And if you find them, we add the antibody and find the antigen. So something indicates that antigen is present here, that antigen is present in our if there are no antigens, it's O negative. If it only clumps with the anti-RH solution, then it's O positive. What is O negative? So we're looking for the antigen on the red blood cell. It will not. If it's not, it won't. Leave it out of our head. Yeah. will not just type your blood, they will do what's called a type and cross match. So they will type your blood, and let's say your blood is O negative. They will go to the blood bank and get O negative blood if you hadn't already put some of your blood there, which you usually do if you have surgery. And then before they give you that O negative blood, they will physically mix a small sample of your blood with the blood that they're going to transfuse you with, just to make sure, before they put it inside of you, that it's not going to, maybe it was wrongly typed or whatever, but just to make sure it doesn't agglutinate. The PhysioX has some of this in it. And it really is not complicated, it's just a lot of different things that you can go, okay. Do you think it's Sure. A couple of drops of blood and 2 or 3 drops of the antibody solution. Small amounts, can't tell anything. So yeah, use a couple of drops of blood and 2 or 3 drops of the antibody solution, but you want to be able to tilt it back and forth and see if it's actually agglutinated. And again, it won't form dots so much and it'll get thick and jelly-like, or at least thicker.
So we'll just number these through brown.
Okay, that was Mr. Jones.
Those are on it? I'm gonna put the name next to it. Yeah.
Is that something really developed?
This is,
It gets thicker. Yeah, so for some it's really strong, for others it's not as strong.
So there's no flooding, all flooded A positive.
Somebody else is, okay. Which one is
If there's no flooding,
Okay, what is that? There's no clotting. So what does it say about the antigens that are present? Are there any antigens present? Or the blood type?
When it's clotting,
You get sick, you can look at the microscope under the 3x, you can see a little bit. But, yeah, use the shortest one. You don't have to, you should be able to do it in the tray. If you use the shortest. If not, I can give you a microscope slide. Actually, let me see if we have a dissecting scope.
Yeah, we use the same A in all of them.
See, this one you can tell already.
Use another N. It's an A.
You don't have to do that.
Only in
I was thinking of like three.
It's clumping. It's not clotting, it's clumping.
This one in there.
Clotting is the long part.
So it's for every antibodies. So if our.
So we're not looking at the antibodies present, we're looking at the antigen present.
So we added A antibody, anticoagulated.
Not quite like this.
That means we have B, right?
No.
Is RH?
Again, we're not paying any attention to the antibodies that are present. We're not looking for the antibodies. We're adding external antibodies. And if they find an A antigen, we have an A antigen. Does that make sense? Yeah, this is going to cause that. If it's an antibody, we're not doing this at all. We're adding an antibody, and if it causes the antibody against these, so if it causes agglutination, that means the B antigen is present. So we're searching for, it's like putting a magnet looking for iron.
So we add A such coagulated.
We have it. Everything's good. You're right on that. You're right. Okay, guys, be careful not to call this coagulation. Because coagulation involves fibrinogen and fibrin threads. This is agglutination. Okay, we're not forming blood clots, we're clumping blood cells together. So this is agglutination. You just put it on the table there. Right. They're probably still out.
To be O positive, nothing
Oh, yeah, the trays get washed and put back, don't throw them away.
They're not comprehensive.
Don't forget to quit online
Coordination.
That's what I meant. It's
It says right there. No. So, yeah, that doesn't have any antigens. Any No, see, this is.
So you girls have O negative, A positive, or A positive.
Wow, we're fast, we're fast.
Yeah, to get the idea of my.
For A and RH only?
So if it, what I did, it was doing dots, and if it didn't, then,
It's never enough dogs. You are?
Otherwise, they don't have it prepared for.
all of the
Yeah, so let's try that. Yeah.
Okay, so we want to double check that we have this.
Okay. Uh-huh.
Not the RH,
Correct.
correct? So that RH, we're kind of iffy on that.
This little bit as it flows, you have, this one looks like it has nothing.
Right.
Yeah, okay.
That's what I'm iffy on that.
So, yeah,
Should we do it over again, or?
No, I see a little bit in there. Yeah.
Okay, now for Mr. Smith, I don't understand how we said that ever. So this is coagulation, the RH and not the.
Correct. Okay, so that, you can see that, okay, in there.
Yeah, which makes this.
And this one?
That one.
Okay. And like I said, the key is not so much in figuring out this particular lab, whether it's agglutinated or not, but if you know that that's agglutinated and those two are not, what's the blood type? Yeah. Okay? Yes?
Pretty sure. Well, AB, so that, I mean, with A and RH agglutinated, but
I would say that D is as well. And it's because what you added to it was a different color. But it's still, if they have to look at it, there's stuff in there. So we tell, as I start to run some of the clear, I can look up the names of those Sally. So I would say all three of those.
are they all going to be the same?
Do you have Mr. Brown?
Can you mention the first one?
Yeah? Well, yeah, Mr. Brown is on there, but Mr.
I'm more concerned that what I see you've drawn, you interpret correctly as the right side, and then whether that's.
Because I'm wondering if we're all supposed to have the same answer.
Yeah, they should, I mean, they're supposed to be pretty much, but like I said, that's why I have to draw here. What I check, it's not that this is just, like, let's try to stay here. That should be right there.
What about Mr. Green and Mr. Jones? Is
That
it,
should be two different things.
they should be two different?
Yeah, so what'd you get from Mr. Green?
Hey, do you know that Mr. Green and Mr. Jones
Oh,
should
not
be different?
present. Yeah, B is present.
But there's no images to any.
Oh,
Yeah.
So which one gets the So we know it's either
All of
A positive
All of them.
or AB. But it doesn't have any images. So that kind of is the cell or AB. Any of those has to be A.
Mr. Green has all of them.
Get rid of this one.
I would eat it, but what is that? These are the actual antibodies, and you can use this on real blood, right? And it's simulation.
Blood type,
And these are just, so if you look at it in the microscope, there's going to be little circles that are meeting up together. So
there's
they're
possibilities.
not just like concave.
A, right? Or AB. You're right, but no,
Yeah.
everything.
So,
No,
each of these from here to here, so you can divide it by the
But then we know it's possible.
and that's the same for all three. Okay? So you divide this length by this total length, and this length by this total length. And you'll get a percent. Okay.
So it has to
Not
be.
yes, but it's going to be a possible
Nothing's
cause.
D?
Yeah. Because you wouldn't know the number of what it's actually causing.
So, only, so, B, person B is starting at all.
So if our peaches, if
They're all started at So you're
The other way around. Yeah, and on the last down, I'll show the resistance on A.
Yeah, you don't know.
That one,
Yeah, there's no
No, you rinse it out in the sink and then. Let me revisit
is this, medication?
yeah.
I'm just still getting it straight.
Yeah, fine, here, right?
She actually formulated.